So here's a slide that is basically what kinds of things people do when they're doing genome annotation. They might calculate the GC content. So you have the whole, or you have some part at least of the genome, and then you're saying what percentage of the genome is composed of GC. And you might wonder, why is that even important? Because that's how people classify bacterial species. So you might know that a species is when two individuals cross and they produce a fertile offspring, then they're considered to be of the same species. If they cross, they most likely will not get pregnant, or if they do get pregnant, they may not make it to birth, or if it makes to birth, it will be sterile. So if the two individuals that mated are not a part of the same species. But then you're like, oh, but bacteria, do bacteria have sex? Yeah, there are occasions where there's like a pilus, so a hair that grows and attaches to another bacterial cell, and then one transfers DNA to the other cell. But the bacteria also, they can just split, and they don't need another one to make babies, so new bacteria. So if we can't say that, if we can't think about having offspring that is fertile, then how do you classify bacteria? It's by their GC content. So the other thing people do when they're doing genome annotation is looking for SSRs. This is very useful when you're trying to look at the genetic diversity of populations. I know I did that when I was undergrad. Yeah, when I was your age. I was working with microsatellites. Dr. Steven from OENB works with microsatellites, and we are using microsatellites to try to see if we can tell apart different populations of ferns, for example. And then the biggie is this part that I was telling you. We're predicting coding DNA versus non-coding. So I was referring to it as genes versus non-genes. But there are parts of the gene that is actually non-coding. So you might wonder, okay, what does coding mean? Coding means that codon of DNA is encoding an amino acid. So not every part of DNA will correspond to an amino acid in the protein. So that would be the non-coding. So the coding will produce protein. The DNA is transcribed. So transcribed is DNA is read and an mRNA or any RNA molecule is made corresponding to that DNA sequence. And translated is the mRNA becomes or is encoding protein. Non-coding, there are two different cases, right? One, the DNA is never transcribed. So that is true for intergenic regions, which is DNA in between genes. So that DNA does not get made into any version of RNA. And or it's a regulatory sequence, which is, for example, is a promoter or an enhancer. These are also not part of the gene, but they have to do with gene expression. We'll learn more about this later. And or the DNA, so it's either not transcribed or it is transcribed and not translated. So you could think about, okay, genes are sequences that are transcribed. And so in that case, it is a gene, but it's non-coding. For example, an intron is a part of a gene that gets chopped out and doesn't make it to the protein that corresponds to that sequence of DNA. This is transfer RNA. That's the guy that brings the amino acids to the ribosome. This is ribosomal RNA. So there is in the DNA a sequence that encodes ribosomes. And it's really important because if you can't make ribosomes, you can't make protein and you're not viable. And then there are other small RNAs that people learned about them more recently. And so the other things we do is genome annotation is we predict genes, including the 5' UTR, and you predict within the gene the exons and the introns. So maybe you know what this means, maybe you don't. But here's a graphic that will try to drive this point home about the gene, the parts of a gene. So imagine this black line here is a DNA sequence, okay? And then these boxes that I drew here are the parts that are genes. So there's some gene here, some other gene here, another one here. They don't have to be the same length as you see. The part in between the genes, we call them intergenic DNA. This DNA sequence that is upstream, so I drew this as the 5' here, the 3' here. So whenever something's before, we call it the upstream. So the sequence upstream of this gene this sequence of DNA here has some cues on when and how much to transcribe this sequence. So that sequence is called a promoter. It's a DNA sequence that promotes transcription. So we have some gene here, some other one here. And then let's focus on the one in the middle because I labeled the parts of a gene. So upstream is the promoter, which is not transcribed. So only the part that is in a box is the part of the DNA that gets transcribed. So there's an RNA version of this DNA sequence. So the part that is transcribed is the 5' UTR, which is the untranslated region. So this part is transcribed, but it's not translated. So it's part, this box here that's DNA right now, there will be an RNA version of it that goes from here to here. But the parts that will end up in the protein, so the codons that will be read and end up in the protein will start with the start codon. That's the ATG that you guys looked in the sequence. Then there's a bunch of other sequence that is specific for every gene. Sometimes you have these introns, sometimes you don't. So the intron will be a sequence of the DNA or a sequence of the mRNA that will be chopped out and will not be present in the protein. Then there is, okay, some more sequence here. Then here's where the protein stops growing. And this is some other mRNA sequence that does not get translated. You're going to see this when we do gene prediction. And the genes don't always have to have introns. The number of introns varies for every gene. If you are a prokaryote, if you're a bacteria, you don't have introns. So that's a big difference, for example. You'll see later too, but I'll tell you that bacteria, the genes, they're closer together too, because they have smaller genomes, so they don't have the luxury of spacing the genes as much as eukaryotes do. Okay, so sometimes you see this thing called open reading frame. So sequences 1, 2, and 3 are the same sequence, but it depends on in which frame you're reading. So the first one might actually be a protein. Well, peptide is pretty short, just to fit in here. The second one, it's the ATG is pretty close to this PAA, so start and stop. This one is less likely to be a gene. So out of these 3 open reading frames, the most likely one to be a gene is the frame 1, because the start codon and the stop codon are further from each other. And sometimes you see this coding sequence. You saw this when we ran BLAST. So that's coding DNA sequence. And that's the sequence from the start codon to the stop codon, including the introns, because it's whatever sequence ends up as an amino acid in the protein. So you saw this earlier, and I said, okay, let's see where are the genes here. I'm going to ask this as a poll, and I'm asking, could the ATG in base pair number 56 to 50 or to 59? So I'm talking about this one. could that one be a start codon of the gene? And the one at 72 to 74, so this one, do you think that this would be the start codon and this would be the stop codon of a gene?